Assalamu alaikum viewers. Today we will discuss about the DNA replication in case of the prokaryotes. So the DNA replication is a process through which DNA is being duplicated. DNA is being duplicated in case of both prokaryotes as well as eukaryotes. There is a little difference between the replication of eukaryotes and prokaryotes. We will discuss later on. So the first part we will discuss about the replication in case of the prokaryotes. We know that a DNA is made up of nucleotides. The basic units or the monomers of the DNA are the nucleotides. Every DNA strand consists of the nucleotides. So what are nucleotides? Nucleotides are simply the monomers that are made up of a phosphate, a sugar, and a heterocyclic base that might be purine or the pyrimidine. So you can see here the DNA is a double-stranded, or we can say DNA is a double helix that run anti-parallel. What anti-parallel means? The one strand that is in the DNA is 5 to 3 prime and the other strand that is opposite to the first one. If first is 5 to 3, the next one is 3 to 5. This is known as anti-parallelity of the DNA strand. So the thing that we are going to discuss today is replication. What is DNA replication? Why replication occurs in case of the prokaryotes or eukaryotes? Today briefly we will discuss about the replication in case of the prokaryotes. After that we will look into the replication in case of the eukaryotes also. DNA replication is the process that can duplicate the DNA of a cell. Means that when an organism is going to divide, it divides its genetic material or the DNA before the division. Before the division, it ensures that its genetic material is being divided equally into its daughter cells. DNA replication answers to the question, when a cell divides, it needs extra DNA. It needs extra DNA so that the extra DNA is being equally distributed in their daughter cells. Where this extra DNA comes from? There is machinery inside the cell. That machinery is being utilized to duplicate this genetic material as being distributed in the daughter cells. Arthur Kornberg compared DNA to a tape recorder or tape recording. That means exact copies can be made from it as from a tape recording so that this information can be used again and elsewhere in time and space. DNA replication, we have learned about the cell cycle, how cell cycle occurs, when DNA replication occurs in cell cycle. So the DNA replication, how this DNA replication occurs, the answer to the question is there is a mode of replication that occurs in case of the prokaryotes or in case of the eukaryotes, that is the semi-conservative mode of DNA replication. Look at the structure of the DNA, double helix. There is a base pairing here and there are broken hydrogen bonds that creates a replication fork, and the strands which are shown here are the leading and the lagging strand. Is the leading strand and that is the lagging strand. Okay. So the semi-conservative mode as proposed by the Watson and Crick, the semi-conservative mode of DNA replication in case of the prokaryotes is being proposed by the Watson and Crick. And this is not the only DNA replication model. There are other models also. There are three models. The first model is the conservative model. The conservative model says this blue color is the parent DNA. Here in one line, these are the upper one line, these are the parent DNA strands. Here you see in first model, parent DNA is being completely conserved as it is. Okay? And new DNA is made from it. And new DNA is made from it. In the next model, you see that parent DNA is as it is, and from one strand, new strand is made, and from the other strand, another new strand is made. So one of the parent strand is being conserved, and that is why this is known as semi-conservative. That means half of the parent DNA is being conserved in this type of replication. Another is the dispersive. You see, in the parent DNA gives rise to a kind of DNA that contains bits of new strands and bits of parent strands. And this model is known as the disperse. That means new strands are made and these are dispersed in the parent strand or spread in the parent strand. But this is known as the dispersive model of replication. Now, we know that the DNA replication that is being most accepted model of the DNA replication is the semi-conservative mode of DNA replication. So according to this model, semi-conservative model, DNA replication would create two molecules. DNA replication would create two molecules. Each of them would be a complex of an old parental and a daughter strand. Parental and a daughter strand. See, semi-conservative. If we see the semi-conservative, here this is the parent strand, and the new strands that are being made from it are one is parent strand and another one is the new strand. The conservative model of replication says, according to this model, the DNA replication process would create a brand new double helix made of two daughter strands while the parental chains would stay together. See, here the parental DNA or parental strand, more appropriate is, the parental strand remain completely conserved. Parental strands remain completely conserved, and new daughter set or two daughter strands are being made. Another is the dispersive model. According to this model, the replication process would create two DNA double chains, each of them with parts of both parents and daughter molecules. As seen in case of the dispersive model, the bits of parental and the bits of new strand are made from the parent strand. This is a dispersive model. The most accepted model in case of the replication is the semi-conservative model of replication. Most accepted model is semi-conservative model of replication. Now we know the replication in case of the prokaryote starts from a single origin that is known as ORIC in case of the E. coli. The real sequence in case of the prokaryotes is mentioned here. This sequence is 80 rich sequence or 80 rich regions. So some regions are called the DNA box regions. Here DNA DNA A box regions is here. DNA A, this is the specific sequence from where the DNA replication starts. Now we are going to see the initiation of the replication process. The origin of replication, we know that this is called the ORIC, origin of chromosomal replication. What are the important features of ORIC? The first thing you need to remember, it is the AT rich region, or there are some regions, those regions are called DNA A boxes. Why? Because there is a protein called, or there is an enzyme called a DNA A protein. The DNA A protein has affinity for this box. This box you see here, this is the DNA box, DNA A box. The DNA A protein has affinity for this region also. See, initiation of replication starts from the ORIC, that's known as the origin of replication. These are the DNA A box, this is the AT rich region. So DNA replication is initiated by the binding of DNA protein to the DNA A box sequence. Means these are the DNA A box. This is the DNA A box, this is the DNA A box, this is the DNA A box, this is the DNA A box. DNA A protein has the affinity for the DNA A box. That means this DNA A protein is going to bind with the DNA A box. When this DNA A protein binds this box, it creates a topological stress. It creates a topological stress. That topological stress would affect this AT rich region and cause unwinding or breakage of the hydrogen bonds in AT rich region, this region. That means unwinding does not start from this region, DNA A boxes. Unwinding starts from the AT rich region due to the topological stress caused by the DNA A protein when it binds to the DNA A boxes. See here, you need to see here. So this is the DNA A protein. When it binds to the DNA A box, it creates a topological stress in AT rich region. That AT rich region is opened up due to the breakage of the hydrogen bonds. So cause the region to wrap around the DNA A protein and separates the AT rich region. Hope you understand this. DNA B protein, that is called the helicase, binds to the origin. Another enzyme that is known as the DNA B protein, that's also called the helicase, that binds to the origin. This helicase enzyme, it uses ATP for its energy. Helicase separates the DNA in both directions. This creates the two replication forks. Because we are talking about the replication in case of the prokaryotes. The replication is proceeding bidirectionally from both the directions. So that is why we are using two helicases. Okay. See, this is one helicase, this is another helicase. Both are in the two directions. So this is the replication fork, and when this replication or when the separation of the two strands creates a single strandedness, this is somewhat unstable. Somehow unstable, and this instability is being eliminated, or this single strand is being stabilized by another protein. That protein is called the SSB protein. That is known as the SSB protein.